going to get into Chapter 2, which is reiterating cell structure and function. Some of this will be a review, but the same caution as we gave last week. Even if it is a review, I still want you to go through and become very comfortable with the role of different organelles and cell structure, because we're going to put everything to practice this semester. So just as I'll try to do all semester and outline each of the objectives for each chapter, again, these should be located at the front matter of the chapter. We're going to talk about the structure and function of different features of a cell, including the cell membrane, nucleus, ribosomes, rough ER, smooth ER, Golgi apparatus, mitochondria, lysosomes, peroxisomes, and our cytoskeletal proteins called microtubules, microfilaments, and intermediate filaments. Okay, so what that means is we're going to review your handout number 1, so maybe you want to get it out too while we're here. Second objective is we're going to conclude, and we won't get to it today, but next time we'll differentiate between proteins which are synthesized for use inside of the cell, like making the cytoskeleton or enzymes or whatever, or to be secreted to act as like hormones or neurotransmitters or whatever the case may be. The front matter of this chapter starts out with a review of our macromolecules. I will name them today, but again, if you're deficient in the background, or if it's just been a while, I need you to read the entire front matter before getting into the organelles. So we'll talk about the different macromolecules. You need to review transcription and translation and genetic information following transfer following cell division. So we won't go into transcription, we won't go into translation, but we're going to say, well, when they happen, these proteins, and what do they do, etc. So we're going to put it into use. To review our major macromolecules, we have 4 classes of these, and you'll see these at least mentioned in each of your biology classes, or you might start seeing a theme in your classes that you take. The 4 major classes of macromolecules include carbohydrates, and what are those?
Sugars.
Sugars, basically, right? Complex sugars. We also have lipids. We also have proteins, and we also have nucleic acids like DNA and RNA. Each of these molecules make a contribution to a given cell. So carbohydrates, as we see, have something to do with nutrient storage and nutrient use. But we're going to study today, they also are components of cell membranes. Same thing for lipids. They also could be used for energy or nutrient storage, but they're also very critical for cell membrane construction. And the exact same thing applies for our proteins. Nutrients, cell structure, membrane structure, right, function, messaging, lots of functions capable through the use of proteins. And of course, our nucleic acids. You may not be familiar with the structure of DNA or RNA, but you know it has something to do with genetic transfer and coding for proteins, for example. So we will talk about different types of macromolecules. Before we get into specifics of a given cell, we need to go back and revisit what we kind of introduced last week about a thing called the cell theory, a scientific theory called the cell theory. If you remember, we went through the hierarchy on Thursday. We gave a definition that cell is the basic unit of life. And we said we're going to support it today with the cell theory. There are 3 major tenets to the cell theory, and there were originally 2, and then they were followed up with another in addition. So there's 3 tenets to the cell theory. The first is that we used for our organization. Cell's the basic unit of life. Reminding you that this is a theory, not a scientific law, so that not everybody supports the cell theory. And you don't have to outside of class, but for classroom and making this class cohesive, this is what puts it together for us. Okay? So we're going to go with the cell theory, that the cell is the basic unit of life. Furthermore, when we look at multicellular organisms such as yourself, the repeating unit of any living organism is the cell. Those 2 might seem redundant, But if I go with the kind of an extreme, silly example, like take, for example, the pin on your hand, at your hand, it has some of your epithelial cells, and it's got however many billions or trillions of microbes on it. Is the pin itself living? That's trillions of cells on it. Why isn't it living? The pin itself is not made of cells. It's made of some plastic polymer or something like that, which is not living. It's not a cell. So in order for something to be considered living, its repeating structure is cellular. And then the third tenet which was added is that cells come from pre-existing cells. And so cells must divide to produce new cells. All right, so those are the three tenets of the cell theory. We're going to support this class with that cell theory. What I want to do now is look at some various cell structures which you would find in a given eukaryotic cell. This image I like to start with because it's super generic. You've probably seen it. Same one I think I have from your handout. I don't like it. It says oversimplified, stripped down of any specificity eukaryotic cell. So it's super generic. So up till now, all of your cells in your textbook have looked boring, basically. What I want you to be aware of is that your cells are not boring. In fact, they're very complex and busy living beings, right? Cells, if you will. Inside of a cell, you will find the familiar organelles, like for example, mitochondria, nucleus, the rough ER, the Golgi, the smooth ER, and etc. But I like this image that I found last semester, that it helps us put it together that our membranes are more than a flat line in a textbook. And they're more than maybe the phospholipid structures that you've seen before. Yes, the phospholipids are there, but there's also proteins incorporated into the cell and having different structures and different functions to contribute to the cell's homeostasis and what it does, what its contribution is to the whole body. What kind of cell is this? Does anybody know? A nerve cell, like you might find in, say, your spine, coming out of your spinal cord, for example. All right, so the membrane is complex, okay, as well as not only do your membranes have proteins, your internal organelles also have membrane proteins, not just phospholipids making up their membranes. So let's look at our cell membranes. And the image that I have shown is not the exact one in your lab manual or your handouts. Can y'all work between the two, or do you need me to put this image up? Can y'all work between the two, or I'll go slow so we can walk through it. But I'm going to use the image and the labeling on your guided notes to kind of go through this. This one's nice though because it's colorful, so it's easier, but nicer to show. In a cell membrane, we're going to have three major components. What are the three major macromolecules which you might find in a cell membrane? We have what? Okay, we got proteins, lipids, and two types of lipids. So let's break one down. So proteins, we have proteins. What kind of lipids? Phospholipids and cholesterol. Very good. So in the margin over there by your textbook, let's put, or by your guided notes image, let's put these three things. Who did this? All right, so we're going to start over. Our membranes are going to have proteins. What was also mentioned? Phospholipids and cholesterol. In any cell membrane, you're going to have a mixture of proteins, phospholipids, and cholesterol. The type of proteins, the type of lipids, the type of sterols are variable depending on the cell type, right? If you're looking at a cell in your brain versus a cell in your heart muscle, okay? But or the plasma membrane of, say, a red blood cell. But you're still going to have a mixture of those. What are the purple-colored things, the darker purple-colored things here? Representing some type of what?
Glycoprotein.
Just any of them are
internal membrane protein.
A protein, some type of protein. Okay, so those dark purple things are types of proteins. The lighter colored things are actually, we didn't have a list, but they are what? The phospholipid head will be the blue. So what about the light purple stuff?
Glycolipids.
They're carbohydrates, sugars. Okay, so we'll come back to these. And so there are carbohydrates on some proteins, there are carbohydrates on some lipids. But the phospholipids are these little spear-looking, balloon-looking things on here. So in this image, the blue spear things are the phospholipids, arranged in what we'll call a bilayer. Anything in this darker purple colors is just a protein. We don't know anything about it yet. And in the lighter colored things are carbohydrate groups or sugar groups. The bright yellow is cholesterol, very good. And this tan area is the cytoplasm, and so intracellular fluid, so these little filaments in there are not part of the cell membrane. Okay, so I'll come back to that. Does anybody know what they are? Cytoskeleton. Okay, we'll address that later. But let's go to your image, this kind of black and white simple image. Let's go through it in order. Item A is pointing to a what? A single phospholipid, and then A1 and A2 gets into the details of it. So let's just call that a what? Let's call it a phospholipid. And if we look at its phospholipid structure, we notice it's kind of complex. And then we can see labeled A1 and A2. A1 is referred to its what? The head, and the A2 is the tail. The phospholipid head and the phospholipid tail. So A1 labeled as the head, and A2 as the tail. What features do we know about phospholipid heads? The hydrophilic. Hydrophilic. H-Y-D-R-O. Philic. P-H-I-L-I-C. Hydrophilic. Heads are hydrophilic. What does that mean? Water loving. Water loving. Do they really have love? What does that mean?
Attraction.
Attraction. What kind of attraction? We'll say electrical, right? Electrical attraction. So something that is hydrophilic is attracted to water. And so really we could say it's what? Yes, it's hydrophilic. We could also say it's polar. P-O-L-A-R. The head is polar. It means it's water loving. It can dissolve. It's water soluble. The phospholipid heads are water soluble, they're polar. They're attached to some fatty acid tails, or chains called tails. What do we know about these phospholipid tails? Hydrophobic. The tails are hydrophobic, meaning water fearing. They're not attracted to water, they're non-polar. So the tails are non-polar. So item A, we identified as a phospholipid. Then we said A1, the head is polar, water soluble, and the tails are non-polar and not soluble in water. we okay to move to B? B is pointing to that whole thing, that phospholipid and the things attached to it. What is the thing attached to it? Sugar, you could say a carbohydrate. It's not a single, it's like a monomer, so I would call it a carbohydrate. So I have a carbohydrate bound to a phospholipid. What do I call those?
Glycolipid?
Glycolipid. Label item B, that whole collective thing, the carbohydrate affixed to the phospholipid. Label that a glycolipid. Glyco, glycolipid. So an example of a glycolipid in this image would be the carbohydrate attached to the lipid.
Oh,
When they're bound together, it's a glycolipid. Yes.
I thought the ones that are attached to the
To the protein? What do you think we would call a carbohydrate attached to a protein? If we call a carbohydrate attached to a lipid a glycolipid, what do you think we call a glycoprotein? So this thing here would be a glycoprotein. What image on your handout would represent a glycoprotein? Item C in your handout, or not the handout, but your guided notes. Item C, I would label as a glycoprotein. And item D specifically is referring to what? The carbohydrate group on there.
So these are, this combined with this, the glycolipid and sandwiches?
That would be yes, this is a glycolipid and this would be a glycoprotein.
So,
It's like the whole.
Yes. Yes.
so the glycoproteins, like they're only gonna be like the little tree things that are attached to the protein.
Yes.
So they're not like any of like the purple stuff underneath.
Anything here, for example, this protein here and this protein and this this protein, this protein, this protein, these proteins, this one. If we cannot see a discernible sugar carbohydrate attached to it, it's just a protein.
Right. Oh, so is the whole thing like the little tree, that whole thing is the
The
glycoprotein?
whole thing would be the glycoprotein because they're bound together. Glyco refers to sweet, so it's talking about the carbohydrate, and the protein would be the protein. So when they're complexed together, you would call it a glycoprotein. If I see a carbohydrate attached to a lipid, I would call it a glycolipid. But they're bound together, one whole molecule, but part of it's a sugar, part of it's a lipid or protein or whatever it's affixed to. Okay. Does that make a little bit of sense? And that's a good question. Glycoproteins and proteins are not only just part of a cell membrane. They're inside of cells, they're outside of cells, they're hormones, neurotransmitters, right? But we're looking at them exclusively in a cell membrane at this point. So item C in the handout is representing a glycoprotein. Item D is looking at just the carbohydrate group, and we could have pulled off the carbohydrate group on the other side. On B as well, called it a carbohydrate. Please notice though, carbohydrates face, when they're associated with a lipid or protein, they face externally. So they're facing the extracellular fluid. So in this image, where can we label the extracellular fluid? On the top, right? However you want to orient that, right? So back to H. H through I think, is representing the bilayer itself. So H and above, I would refer to as the extracellular fluid. The fluid environment on the outside, outer top in this image. Extracellular fluid. And therefore, where A, G, F, E, and E, right, are located, you would label that the what? The what? Intracellular fluid.
So is the cytoskeleton always gonna be on the inside?
The cytoskeleton will always be inside of the cell. Yes. It may affix to the membrane, but it's not part of the membrane. It doesn't make the membrane itself. D is just a carbohydrate. All right, what about item E, squished over there, representing a cholesterol? Item F, a different type of protein, and I'm gonna add some detail to that in just a second. Item G. Y'all are correct, but it's also a type of protein. So let's add that. You guys are already going there. We called item GH. Some people mention it as a channel protein. And notice that its structure, it looks like a channel. It spans how many layers of the membrane? Two. So there's a continuous channel through the cell membrane connecting which fluids? The intracellular fluid and the extracellular fluid.
Would this also be a channel that like waste would follow through, or just have a fluid?
Depending on the type of waste, what we're going to learn is how something gets across the membrane depends on its size and solubility. And so probably not waste getting through there. Usually you see ions or water carried through channels. Okay, so if you want to make a little arrow, you put it bidirectional, it's always open. So something can simply or facilitate through that open channel. And so if this were my channel here, or this one's even closed, but something could diffuse through into the cell, or something could even diffuse out. And in chapter 4, we'll look at the details of what would go through that or not. So the detail that I wanted to add is that some proteins are actually incorporated into the membrane and some are not. Okay, so the term I want to differentiate those that are incorporated and those that are not would be integrated or integral versus peripheral. Integral versus peripheral.
That's not there.
I haven't labeled it yet, so we're just setting you guys up. A protein, when associated with the membrane, would either be integral or peripheral. Integrated, incorporated in the membrane, or at the periphery, at the margins of the membrane. So let's look at our proteins. Item G, how could we classify that channel protein? It is it's integral. Okay, so item G, yes, it's a channel, but because it's a channel, it's got to be integrated into the membrane to make that channel. What about item C? Protein C. It's integral. It doesn't go through two layers, but it is in one layer. So it's integrated into the membrane. Just because a protein is integral to a membrane, it doesn't have to span both layers. Well, I'll start on that. This is integrated, for example. So item F would be an integral protein as well. There is one peripheral protein here, but it doesn't have a label. So the bottom lower left-hand side of your image, you see the little circle thing. That's a protein. What kind of protein is that? Peripheral. And let's draw one facing the extracellular fluid as well. Squish in an item K. Somewhere up there. And call that also a peripheral protein, but it's facing which fluid? Extracellular fluid. So your peripheral proteins may be extracellular facing or intracellular facing, but they do not do what? They don't span the membrane at all. We'll conclude class here. Go ahead and read chapter 2, right after, I think section 2.6 and on, so that our discussion on Thursday will go more quickly. And don't forget your handouts will be due on Thursday as well. Question? Yeah.
They last to me like
I would just call it integral protein. Could be an enzyme. I don't know. Generic. We'll see you guys in lab or in class. If you are late, I might want to sign in.